So today we're talking about the nervous system. This chapter is always interesting, I think, for this class because for some of you, you know this already. You've taken all these classes already. For others of you, this is brand new. You know nothing. And then, of course, there's all sorts of people in between those two. All right? So I always say this. If you know nothing, try to stick with me. Feel free to review things in your book. Feel free to look things up, learn them more. Don't get too overwhelmed, though. This is not the most important of all the things we talk about, okay? But you do need to know it. If you already know all of this, hang in there with me. Hopefully this will be an easy chapter for you, okay? So, with any drug that we put into our body, food, drug, anything really, right, it changes our internal environment, okay? Our bodies like what we call homeostasis, right? I feel cold, I put on my jacket so I feel better. I feel thirsty, I drink water so I feel better. I like my body temperature to stay about the same, my water content, my glucose concentrations. We like our internal environment to stay within these kind of limits, right? Now, psychoactive drugs influence homeostasis. They disrupt homeostasis. Now, if we use that drug on a regular basis, okay, as to where it was messing with homeostasis before, every time we used it, if we use it on a regular basis, our body starts to think that's normal, okay, and our body changes its idea of what homeostasis is. Now, if we drink alcohol every day, our body thinks having alcohol is normal, which then means when we stop using alcohol, our body freaks out because it doesn't like being away from what it thinks is normal or homeostasis, okay? So, for people that are not drinking so much, alcohol inhibits vasopressin release, okay, which is an antidiuretic hormone. Basically, this means you drink alcohol and you have to pee more, okay? So after drinking, more urine is produced. Now, this is for light drinkers. When light drinkers or new drinkers drink alcohol, they have to pee more often. Heavy drinkers, on the other hand, do not, because their body is used to that. So it has changed to think that having that alcohol is normal, okay? Now, then when they have alcohol withdrawal, they exhibit this increased vasopressin release, okay? Because they think this is normal. Without it, your body is, I always like to use the term freaking out, okay? So, we maintain homeostasis as much as we possibly can. When we're getting drugs on a regular basis, our body starts to think that is normal, and then it doesn't like it when you don't use it. That's withdrawal. Okay. Now, when we talk about the nervous system, we're going to talk about two major types of cells. We're going to talk about neurons and glial cells, okay? And people talk about either glia or glial interchangeably cells, okay? So our neurons, these are our primary elements of the nervous system. They analyze information, they transmit information, okay? Within the neuron, now I use my hands to kind of talk about what the neuron is here, right? So this is my example, is the cell body. I always think of the cell body as like your hand, that palm of your hand. Okay, the cell body, it contains the nucleus and other sustaining substances. The dendrites would be like your fingers here. The dendrites contain these receptors that respond to chemical signals, okay? Psychoactive drugs activate or inhibit the neuron based on the type of receptor that it is activating or inhibiting. Okay? Now the axon would be like your arm here. The axon conducts what we call an action potential. The action potential happens when information comes in, information meaning neurotransmitters, come in through the dendrites, they collect in the cell body, and when there's enough in the cell body, this information, or neurotransmitter, shoots down the axon and then out the axon terminals, or the terminal button, or the synapse. Then it's hanging out in between. In between cells is called the synapse, or the synaptic gap. And then from there, it is either sucked back in or sent on to the next neuron. The idea is the information is sent from one neuron to the next neuron, to the next neuron, to the next neuron, to the next neuron. Okay? Psychoactive drugs can either inhibit this, making it not happen as much, or can make it happen more, excite this action potential happening. Okay? So here is a picture that is not just my arm, but here we have, what's going on with my pointers here? Strange. Sorry, I was looking for a here. So here we have, the cell body, the dendrites here, the axon, as I was mentioning, information goes out, and out here into this area that we would call the synapse, or the synaptic gap right there. This stuff that's on it, this myelin sheathing, that is this stuff that's here on it, it's kind of like a wax coating that makes information go faster through the axon. If this was a neuropsych class, I would spend a lot of time on this. It is not. You do want to know these things. You do want to know the different parts of the neuron. You want to know the dendrites, the cell body, and the axon. Okay? You do kind of want to know how this works. But you don't need to really intensely know it. Okay? Hopefully that helps. Now, then we have our glia, or glial cells. Now these, one of the big important parts of this is that they create something that we call the blood-brain barrier, and that's one reason this is so important to this topic of psychoactive drugs. Okay, now, going back, major function of these, they provide firmness and structure to the brain, they get nutrients to the system, they eliminate waste, they form the myelin, they communicate with other glial neuron cells. They're kind of like, they're the worker cells, right? They do all the work, all that extra work. So the blood-brain barrier, if you think of kind of a wall that is created by glial cells, and if you sort of imagine, and this is sort of a pretend way of imagining it, but if you imagine drugs going up and then hitting the blood-brain barrier, they wouldn't then affect our cognition. They wouldn't affect the way we think and feel. But in order for something to be a psychoactive substance, it has to be something that can pass through the blood-brain barrier. That is, they call it lipid-soluble. It can pass through the blood-brain barrier. Okay? Drugs that hit you faster, or drugs that are more intense, pass through that blood-brain barrier easier or faster, or both. Okay? Now, I mentioned this before, but this action potential. So within neurotransmission, okay, you have this action potential, which is sending the information or the electrical signal transmitted along the axon and onto the next neuron. All right? I don't need you to know all of these next words. I do want you to know that the action potential is a all or nothing or all or none event. So that means that as the neurotransmitters collect in the cell body, if there are enough, then the action potential happens. If there's not enough, it does not happen. Okay, there's not like a little one. It either happens or it doesn't happen. All or nothing. Okay. Let me just go back for a second. So based on that, the psychoactive drugs that affect these things can either make it so that there are more action potentials occurring or have an inhibitory effect where it blocks maybe some of the neurotransmitter from getting into the cell or something like that. There are like a million different things that different drugs can do, but maybe it does that, and so it makes it so that the action potentials are not occurring as typically they would. So it's either making them happen more or less, but this action potential has this all or nothing thing. Okay. The nervous system. Now, the nervous system, within it, we break it into these different branches. Okay, so we have the somatic nervous system. This is sensory information to the central nervous system. Okay, and the motor information that comes back out. Okay, so sensory information in, motor information back out. Basically, the somatic nervous system is involved with voluntary action. Okay, so I want to move my arm, I move my arm. Now, the autonomic nervous system is different. The autonomic nervous system, if you think of the word automatic, right, autonomic is involved in involuntary bodily functions, like the beating of your heart or your breathing, right? You don't have to remind yourself constantly to make your heart beat. Thank goodness. It is an autonomic or automatic, right, thing that's happening, okay? Within our autonomic nervous system, we have something called the sympathetic branch and the parasympathetic branch, okay? We'll talk about these more in a minute. And then finally, of course, we have the central nervous system, which is just the brain and the spinal cord. So the central nervous system is just the brain and the spinal cord. The somatic and autonomic are kind of everything else. All right. Now, as I talked about before, saying it again, the somatic nervous system controls voluntary actions. It carries sensory information into the central nervous system. It carries motor or movement information back out. So move arm, that was hot, don't touch that. You know, move your hand away from that. Acetylcholine is a neurotransmitter at muscular joints. So it's important in that. So if we have issues with acetylcholine or drugs that affect acetylcholine, it will then affect our neuromuscular abilities. Okay. Now, our autonomic nervous system, as I mentioned before, it breaks down into two branches, the sympathetic branch and the parasympathetic branch. Okay? The sympathetic branch is involved in something we call the fight or flight response, and that is it is involved in increased heart rate, increased breathing rate, increased feeling of I need to punch something or I need to run as fast as I possibly can away from said thing. Okay, so the sympathetic branch, I always think of like if you ran into a tiger, your sympathetic branch would be activated, your heart would beat faster, you would either punch that tiger or you would run from that tiger, right? Okay. The parasympathetic branch is more like a tiny house kitty cat, right? Typically, tiny house kitty cats make people feel calm, and their decreased heart rate and decreased breathing rate, and they're like, Oh, kitties, right? Some people don't feel that way. However, tigers versus kitty cats, right? Sympathetic tigers, parasympathetic kitty cats. Okay. These act in opposition. Okay. Norepinephrine is involved with that increased heart rate. Acetylcholine is involved with that decreased heart rate. Okay. Now, they act in opposition. So when one is affected, the other is chilling out. When one is chilling out, the other one is affected. Okay. Many psychoactive drugs affect the brain and the autonomic nervous system in some way, right? They are making us, I mean, this is why they're called like depressants or stimulants, right? Okay. All right. Central nervous system, brain, spinal cord, it helps with integration of information, learning, memory, coordination of activity. Clearly important, okay? All right. So let's talk about some of the chemicals that we find in the brain. And these would be our neurotransmitters. Dopamine, okay? So dopamine is going to be part of this, you know, the well, what do I want to talk about? Yeah, so the mesolimbic dopamine pathway. It's proposed to mediate psychotic behavior. So dopamine is associated with like schizophrenia, okay? So on one side of dopamine you have schizophrenia, on another side of dopamine you have Parkinson's, okay? So that would be kind of interesting. So Parkinson's is more of a neuromuscular, people end up having shaking and difficulty moving with Parkinson's, as to where with schizophrenia we're talking about more of psychotic thinking. So since these are two kind of ends of the spectrum of dopamine functioning, if we have somebody who has schizophrenia and they're given medications to stop some of the psychotic symptoms, if they're given too much medication, they end up with what we call Parkinsonian-like symptoms, right? They have symptoms that look like Parkinson's. On the other hand, if you have somebody who has Parkinson's and they're given kind of too much medication for that, Parkinson's, then they end up with schizophrenia-like symptoms or pseudo-psychotic symptoms. And we notice that when the medication is then, the dose is lowered, those psychotic symptoms go away if it's a person with Parkinson's. Okay, so we do see them working on that dopamine pathway. Okay. Dopamine is also associated with that component of the rewarding properties of drugs. So dopamine is often talked about when we talk about, like, why do you keep taking that drug? Well, because it's getting some part of the brain that feels really good. And it's rewarding to keep doing it over and over and over again. Okay? All right. This is just a picture of our dopamine pathways. Now let's talk about norepinephrine. Okay? So this is arising from the locus ceruleus. These are things that you read 100 times and you go to say them out loud, and I don't know how. Okay. In the brain stem, and then projecting to multiple brain areas. Norepinephrine regulates the level of arousal and attentiveness. It seems to play a role in initiating food intake, meaning appetite. Serotonin. Serotonin is going to have multiple pathways arising from the brain stem, that raphe nuclei, and then projecting out to multiple brain areas. Serotonin does seem to be related to impulsivity, aggression, depression, and control of food or alcohol intake. So basically not being able to or being able to control your eating, or your alcohol. Okay. Also, serotonin is definitely thought of when we think about depression, which is why a lot of our depression medications are SSRIs, or selective serotonin reuptake inhibitors. We'll talk more about that during the section where we talk about mental health. Okay. Hallucinogenic drugs like LSD, for example, do tend to influence serotonin pathways. Interestingly, if somebody's been taking medications for depression, so SSRIs, for a long time, and then they go to use like LSD or psilocybin or a classic hallucinogen, sometimes they don't see an effect from that because they've been using the SSRIs for so long. They then are not affected by classic hallucinogens, which is interesting. Okay. Acetylcholine is rising from the nucleus baleus, projected widely throughout the cerebral cortex, right? It's involved in Alzheimer's disease. So acetylcholine is often talked about when we talk about learning and memory and Alzheimer's disease in general, okay? Now, this is a little different, but endorphins, right? Endorphins are found throughout the brain. They're naturally occurring opioid-like chemicals, okay? Endorphin basically is the word endogenous, meaning like natural, endogenous, and the word morphine, right? Endorphin, endogenous morphine, basically your own morphine that you're creating, okay? They call them endorphins. They play a role in pain relief as well as a lot of other functions, okay? Then we have GABA. GABA is generally an inhibitory neurotransmitter. It can inhibit lots of different kinds of things, depending on what we're talking about. Glutamate is an excitatory neurotransmitter, so it enhances the effect of whatever it is that we're talking about, okay? So both of these are found in most regions of the brain. They can do a lot of different things, but generally GABA is inhibiting things, and glutamate is making it happen more. Here's just another way to look at it. This can kind of be helpful when you are studying. Give this a look over. Major brain structures, again, just when you're kind of looking at things. Now, when we talk about the life cycle of a neurotransmitter, okay, neurotransmitter precursors or beginnings of are found just throughout the blood supply, okay? Then we have what we call uptake, right? Selected precursors are taken up by the cells. Then we have what we call synthesis. Okay, this is where those precursors are synthesized into neurotransmitters. And they've done that through different actions of enzymes. Okay? If this is way over your head, just kind of know the general idea of what I'm talking about. Okay? Don't worry about that. Okay? Now, we have some things here. Okay, and that is storage. And that is neurotransmitters are stored in small vesicles. Release. When that action potential arrives, right, neurotransmitters are released into the synapse, right, into that gap here, into this gap. This is our synapse, our synaptic gap. This is where they're released. They hang out here, okay? And then they bind, and that would be, if you look at these kind of like here, 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 you know, where they're kind of in here, they're binding to the postsynaptic or next neurotransmitter, okay, or not neurotransmitter, the next neuron, sorry about that. The postsynaptic cell would be the next neuron. So they're binding in their particular receptor site and going to be moved on to the next. Okay. Now, then you have what we call metabolism. So that would be that once the signal has been sent, the neurotransmitters are removed from the synapse. They may return to the original, neuron, or they may be what we call metabolized, kind of taken away. Okay, so drug actions, they alter neurotransmitter availability in the synapse. Remember that gap. So it could be that they're affecting synthesis, they could be affecting storage, they could be affecting the release, they could be affecting the uptake, or the metabolism of the neurotransmitter. So many different things could be happening depending upon which drug we're talking about. Okay? A lot of antidepressants block the reuptake of dopamine, serotonin, or norepinephrine, or a combination of those things. Okay? Now, sometimes we also have the drug will have a direct action on the receptor. And so that may be that the drug comes in and it acts just like a neurotransmitter, and it mimics it. So it basically goes into the receptor and activates the receptor just like a neurotransmitter would, a specific one. Okay? Or it does the opposite, which is an antagonist. And that is, it may occupy that neurotransmitter and prevent the receptor sites from being activated by that neurotransmitter. Again, it depends which drug we're talking about. All right. Now, chemical theories of behavior. So many attempts to explain normal variations in behavior in terms of like changes in brain chemistry, okay? You can see, we talked about Hippocrates, the yin and yang, all of these sort of historical precedents for something's going on in our brains and in our bodies. Okay. Then we have what we call the monoamine theory of mood. This is a class you need note cards for, just to let you know. If you're going to study, making note cards for these kind of terms is really helpful. So the monoamine theory of mood would be dopamine, serotonin, norepinephrine, but the idea that too little activity in one of these systems can cause depression. Too much activity in one of these things can cause mania. Right? However, let's be clear. No single biochemical theory of mood or drug dependence explains everything. Okay? Not one single one has achieved sufficient experimental support. There is not a single, I'm saying it one more time, there's not a single explanation that covers it all and explains it all. Okay, you have to have a lot of different theories, you have to have a lot of different things at play, likely more than one neurotransmitter, likely more than one thing is going on. Okay? Now, within the brain, not within the brain, but with the brain, we can look at the brain in different ways. One of those ways is through a PET scan. Okay, positron emission tomography, or PET, PET scan. Okay? With a PET scan, we can directly measure brain activity. If you see this picture, it looks like there are places that look like, we call it lighting up. They light up. Okay, so when we talk about different drugs, we can see when people take them that different parts of their brain light up on the PET scan. So we know that it's activating that part of the brain, which I think is really interesting. Or we can see that things are not lighting up, meaning it's not activating the brain, or it is inhibiting activation of the brain in certain areas. Okay. Limitations. There's injection of radioactive materials, and we also don't get information from a PET scan on structure. Okay. So it's basically just brain activity. A PET scan. Now, with the MRI, we're looking at a little bit different here. Okay, so this is actually an fMRI. So an fMRI is a functional magnetic resonance imaging. Basically, an fMRI is a lot of MRIs being taken in a row. Just like we used to take lots of pictures and then, in order to make a movie. Same thing, kind of what we're doing here, right? What's cool is you can see this real-time changes in brain blood flow. Also cool is that we can do it without cutting somebody's head open. So non-invasive. Invasive. Limitations, though, is that there's no information about brain anatomy. I mean, there's going to be limitations with each one of these things, okay? And I'm actually going to stop all of our information here. Like, I'm not going to continue on. But there are these different types of brain scans, right? So we have the fMRI, the PET scans. Do know the differences between those two and what they're really looking at. So the PET scan is going to focus on like glucose levels. The fMRI is focusing more on this changes in blood flow, okay? And I do stop at this point because I've given you a lot of information. Just to kind of go through again, we talked about, actions of drugs. We talked about neurotransmitters, how they worked. Really do know these types of words, this part of the life cycle. Do know the blood-brain barrier. Sorry, I'm going through just to kind of help you out a little bit. Do know the different chemical pathways for these drugs. Do know the difference between the sympathetic and the parasympathetic branch of the autonomic nervous system. Do know the difference between the autonomic and the somatic nervous system and the central nervous system. Do know, what an action potential is. Do know that it's an all or nothing thing. Do know the difference between a glial cell and a neuron. Do know, at least 3 main parts of a neuron: cell body, dendrites, and axon. I think that's generally it. I'm sure there's a few other things in there, but that's, just giving you some ideas of things to focus on. Okay, that's it. I will see you in the next chapter.